So let's go ahead and begin our third lecture in the patterns of inheritance topic here. So we've been talking about in the last two lectures the basics of genetics, some of the players and key scientists that helped establish the foundations, but we also want to keep in mind how those genetics work in families. So as we go into this third lecture here at part 3, what I want you guys to do is establish your family tree. Set up a family tree with 3 generations, okay? So if you have no children, you are generation 3. Or we also can use the designation F2 for that particular generation. So F, and it's usually done as a subscript, but I can't do that here. So F2 is the generation you would belong in. Your parents would then go into generation F1. And keep in mind, this is your parents, mom, dad, but also aunts, uncles, people who are related to you genetically. And let me back up one step. In your generation, generation 3 or the F2, it would be you and brothers, sisters, cousins, etc. People who have direct genetic connections to you. And I'll draw this out. I'll use a notepad and sketch this out a little bit better so you guys get a feel for what I'm looking for here. But definitely start setting up your family tree. And then generation 1, or what we would call the parental generation, is going to be the generation that includes your grandparents. Now, this pedigree, this little family tree here, only shows one set of grandparents. Really, to be accurate, we should have both sets of grandparents on here. So again, I'll show you a family tree. I'll show you how to draw it out and what it should start to look like. But I want you guys to start to put together your own personal family tree so you can start to get a feel for everybody who has a genetic connection to you, who is related genetically. Doesn't matter if they're half brother, half sister. If they share DNA, you want to get them on that family tree. Now, in this example, we don't necessarily need number 5 from generation 2, uncle, whoever that is, because they don't share a direct genetic connection to you. What they do share, though, is a direct genetic connection to your cousins, which that information is helpful because that may help figure out your aunt's information, which could come back and help you understand your father's or your grandparents' genetic information. All right, so the example we're looking at here is what's called a dominant pedigree. The dominant allele expresses the trait, and individuals who express the dominant allele, now it could be homozygous or heterozygous, but they have a dominant variation, we shade those boxes or those circles in. Boxes representing males, circles representing females. So in this family, the grandfather was affected by whatever this dominant variation is. His son and one of his daughters, or his daughter and one of his sons, inherited the dominant allele. The other son, over here, number 1 in this generation, did not inherit the allele. So that individual is recessive. Now this man married a woman who was recessive, but when they produced a daughter, the daughter was dominant. This woman's dominant, her husband is recessive, but both of their daughters are dominant. So this could be something like certain health issues, certain genetic disorders, or it could be just something simple like eye color, hair color, etc. Dark hair is dominant to light. Dark eyes are dominant to light eyes. All different types of traits. So when it says dominant, and it says affected male or affected female, don't assume, oh, that's a bad negative trait. It's something that's harmful. It could just be, like I said, hair color. Nothing negative about dark hair versus light hair. Okay, but start setting this up for your family. So Mendel helped figure this out and establish the foundation of a lot of these basic genetic problems. Okay, so the recessive pedigree we can draw out the same way, set up the family tree. In this example, they actually have 4 generations. But we have generation 1, 2, 3, and they added a fourth. And then they went back in and filled in everybody who is affected by this particular trait. But in this scenario, it's recessive traits that they're looking at. So the recessive individuals are shown to be expressing the particular trait that is of concern, or the trait that we're trying to watch and monitor. So the individuals that are completely shaded in here, this individual in generation 4, both of these in generation 3, they are completely recessive, homozygous recessive. That is how they will express that trait. But we can go through the family and a lot of times figure out who is a carrier, or what we're calling heterozygous. So number 2 here, number 3, number 4, and then number 4 here, and number 5 here. All these individuals that are half and half are heterozygous for this particular trait, or we would call them carriers for the trait. They don't express it. You wouldn't see it physically expressed, but they carry the recessive allele, and that is how they passed it down, or that is how that allele got passed down to the next generation. Now, you will have, and I almost promise you this will happen, you will have some individuals in your family where you just can't figure out their genotype. You can look at them and say, I know your phenotype, I can see your physical expression, but I just can't lock in, are you homozygous or heterozygous? That's going to happen, so don't panic if it does. All right, so keep that in mind as you guys are working with the genetic problems. So Mendel helped us establish the monohybrid scenario. One variation, one trait, two variations. He also established this thing called the dihybrid cross. This is where you have two traits, and each trait has two variations. So in our example here, we're looking at color of the pea seed and texture. Those are the two traits. So color is a trait. We got a yellow seed, we have a green seed. Yellow is dominant to green. But then we're also looking at texture. Smooth versus wrinkled. Smooth is dominant to wrinkled. So when you're working with a dihybrid, keep in mind it increases the number of possibilities because of how the genes can mix and assort and combine as they get placed into the sperm or the eggs of the parents. So like I said, it will increase the number of different possible combinations for the offspring. So we're not going to focus too heavily on dihybrids. I'll save that, or you guys will get to do that and work with that in a later genetics course. But I want you to understand what it is. Mono is one trait. Dihybrid is two traits. And the two traits each have two different variations. Okay, so here's an example of what it looks like. If you were to cross two individuals that are heterozygous for both of their traits, you're looking at 16 different possible offspring. 9 of those 16 happen to be expressing the dominant variations, round and yellow. 3 out of the 16 express the dominant color of round, or dominant texture of round, but only 3 of them express the recessive color of green. 3 of them express the wrinkled texture, but they express the yellow color, and only 1 out of 16 will express the wrinkled and the green, completely recessive variations. Okay, so again, we're not going to go too heavy into dihybrids. I just want you to understand the basics of it. I'm more concerned about you guys being able to work and understand a monohybrid, as well as some of the other examples and types of genetic scenarios that we're going to discuss here. Okay, so with Mendel's work, he helped establish some basic fundamentals about genetics. We talked about segregation a little bit. The alleles split. Another major principle and concept Mendel established is this thing called the principle of independent assortment. Now, this means that the alleles of each gene will split and what they say assort independently. So the alleles for the A's down here in this example rip apart. The alleles for the B's rip apart. And then how they combine, how the A and the B's combined, this is completely what's considered independent. It's pure chance. You have no idea what the possible combinations will be. There's no guarantee that big A will always combine with big A and that's going to be in your sperm, or little a and little a is always going to be in the sperm. That is the nature of what's known as independent assortment. It's just a lot of random chance and independent combining of these particular alleles. Right? So that definitely applies to the monohybrid scenario. Segregation applies to all of the genetics when we're talking about the alleles being pulled apart. Independent assortment, though, is pretty specific to the dihybrid scenario. So now as we work with genetics, we want to work with probability. What are the odds, the likelihood that something's going to happen? And we use the genetics of the parents to help predict possible combinations or genetics of the child. So there's a thing called the rule of addition that we use when we talk about probability. And the rule of addition says that the probability of these two events occurring simultaneously. That's what we're trying to figure out, is what are the odds or likelihood that these two events will occur together, that you're going to inherit a green Y and a yellow Y, or a big A and a little a, or a big B and a little b. And in order to do that, we look at the odds and then we add them together. That's why we call it the rule of addition. So the probability of getting a heterozygote individual in this scenario is going to be 1/2. So we're saying if one parent is green Y, the other parent is yellow Y, green Y, what is the probability of getting a yellow-green child? Well, the probability of getting this green Y from this parent is 1/4. The probability of getting this yellow Y is going to be 1/4. Putting those two together, 1/4 plus 1/4, there's our 1/2 for the probability of getting a heterozygote from these two parents. Okay, so we'll look at this, and this is something I want you guys to be able to be comfortable with, is figuring out what is the probability. What are the odds that you're going to get? Because those odds are what we use to establish the ratios of the Punnett squares. To say, I think the genotypic ratio of this outcome will be. And that's something you're going to need to be able to do on nearly every problem that you have for practice, for the quizzes, and definitely on the exams. You're going to have to work Punnett squares on the exams, show me the square, and make sure you guys can tell me what are the odds or what are the ratios for each of these particular crosses. Okay, so again, we'll get into this a lot more when I start doing the drawing, and I'll actually draw out the Punnett squares and show you guys how to do that in another lecture here. Can't do it on top of the screen. I have to do it as a separate screencast. So, okay, so another thing to keep in mind is what's known as a rule of multiplication. This is looking at what is the probability of two independent events occurring simultaneously. And to figure this out, we multiply our probability. So instead of adding it, we're going to multiply it. So what is the probability of getting a number 3 with one of the throws? Well, if you have a 6-sided dice, it's 1 out of 6. Okay, there's 6 choices. You're looking for the number 3. One of those sides will contain it. So 1 out of 6. The probability of getting a number 3 with 2 dice thrown simultaneously, or 1 dice and then throw the dice again, the probability of getting the same event twice is going to be 1/6 times 1/6, 1 out of 36. So your probability of hitting double 3s in a game of craps is pretty small. Probability of hitting snake eyes, or 1 and 1, is pretty small. It's 1 out of 36. So we'll want to keep this in mind as we start working through, again, the genetics and looking at the likelihood and probability of certain outcomes.